Alrighty, so this is question number 1 for Unit 5. We're going to read through it. I'll show you where the points are, and then I'll answer it. And so it says leucine aminopeptidases, or LAPs, I'll call them LAPs, are found in all living organisms and have been associated with the response of the marine mussel Mytilus edulis to changes in salinity. LAPs are enzymes that remove N-terminal amino acids from proteins and release the free amino acids into the cytosol. To investigate the evolution of LAPs in wild populations, M. udilis researchers sampled adult mussels from several different locations along a part of the northeast coast of the United States, as shown in Figure 1. The researchers then determined the percent of individuals possessing a particular LAP allele, LAP94, in mussels from each sample site, which is in Table 1. Then it says A, on the axes provided, construct an appropriately labeled bar graph to illustrate the observed frequencies of the LAP94 allele in the study populations. B, based on the data, describe the most likely effects of salinity on the frequency of the LAP94 allele in the marine mussels population on Long Island Sound, and predict the likely LAP94 allele frequency at the sampling site between Site 1 and Site 2 in Long Island Sound. And then C, describe the most likely effect of LAP94 activity on the osmolarity of the cytosol. Describe the function of LAP94 in maintaining water balance in the mussels living in the Atlantic Ocean. And D, marine mussel larvae are evenly dispersed throughout the study area by water movement. As larvae mature, they attach to rocks in the water. Explain the differences in LAP94 allele frequency among adult mussel populations as the sample sites, despite the dispersal of larvae throughout the entire study area. And then predict the likely effect on distribution of mussels in Long Island Sound if the LAP allele was found in all of the mussels in the population, and then justify your prediction. And so we'll get our points by, this is construct. And so this will be one of the things. And then it doesn't really tell us anything more than that. And I'll come back to that in a second. And then we have describe and predict. So that's 2 points. We have describe and describe. So that's 2 more points. We're up to 4, and then something going on up here. And then we have 5, 6, and 7, which means that part A is worth 3 points. Now, on the AP exam this year, there is no graphing. So I'm going to quickly talk about what you would do. And so what they look for is the 3 points where you would get that is you would make sure that you have all the points plotted accurately on a bar graph, because it specifies a bar graph. And then you would label your axes, and then you would use correct scale and units. So it's just a basic little graph. It's actually not even necessary to answer B, C, and D. But it's how you would have gotten 3 points on this. But again, there is no graphing, so we're not going to take the time to do them. And so now we will answer it. And so part A is a graph. We don't really need to do. And then B says, based on the data, describe the most likely effect of salinity on the frequency of the LAP94 allele in the marine mussel population on Long Island Sound. And so what you want to look at is up in the little table, it shows you that the LAP frequency increases as the salinity increases. And so then you would just write that. And so you'd say, as salinity, the allele frequency would increase. You could also write, as the salinity decreases, the allele frequency would decrease. Now, you might say, oh, if it has it, it makes it stronger. So if it has the LAP94, it can handle more salt. Now, you need to also think about it that maybe not having it makes them stronger at low salinities. So you could sort of look at it from 2 different perspectives, but we're going to go with, for the answer just for this one, we're going to say that as the salinity increases, the allele frequency would increase. And then the second part of part B says, predict the likely LAP94 allele frequency at the sample site between Site 1 and Site 2 on Long Island Sound. And so this is asking for a number. And so if you go up and then you look at Site 1 and Site 2, Site 1 is 13% and Site 2 is 16%. If you give any number between 13 and 16, you get it counted as correct. And so I'm going to say the likely allele frequency between sites 1 and 2 would be around, and I'm going to just split it and say that like there's 3, and so then that's like 1.5, so 14. So I'll say 14.5% so that's good. And so it's any number. So they're just looking to make sure that you can look at a graph and you can kind of figure out if you were to go between those two little sites, what would it most likely be? And then it's just an estimate. And then C says, describe the most likely effect of LAP94 activity on the osmolarity of the cytosol. So that means how concentrated the salt is in the cell. And so you need to use that to describe it. And then it says, describe the function of LAP94 in maintaining water balance in the mussels living in the Atlantic Ocean. And so the LAP94, it would increase the osmolality or the salt concentration of the cytosol. And so the reason behind that is because it would need to let's see, try to get equilibrium between the inside and outside of the, oops. And that is to prevent bursting or shrinking. Remember we talked about that? So hopefully that of the cell. And so this, if we increase the osmolality, and then it would make it even on both sides of the membrane. And so then it would maintain the cell in a healthy state by preventing water movement. And then, let's see, most likely effect, and then, okay, so we did both of those. So the most likely effect is it would increase, and then we would talk about the water balance. And so then it would prevent the water movement. And then part D says, marine mussel larvae are evenly dispersed throughout the study area by water movement. As the larvae mature, they attach to rocks. Explain the difference in allele frequency among the adults, even though the babies are dispersed throughout. And then predict the likely effect of the distribution of mussels if the allele was found on all the mussels of the population, and then justify your prediction. And so the first thing is, hopefully it makes sense that if the babies are floating around and then they eventually attach to something in an environment with a different salinity, only some are going to survive. And so mussels with the LAP94 allele would survive at high salinity, and those without would survive in areas of low salinity. And then the prediction says the likely effect on the distribution of mussels if it was found in everything. So there would be, if there were all of them had the allele that makes them better where it's salty, then they would have more mussels be in areas of high salinity and less mussels in areas of low salinity. And then to justify that, this is because in the high saline areas, the mussels could control the movement of fluids in order to maintain cell size and shape. Or you could say the opposite of that. The ones with, they would not be able to exchange the, or sort of mess with the osmolality when it comes to when it's in areas of low salt. And so we did all of our parts, so hopefully that helps. That's question number 1.